Welcome back to Lecture 2 of the nervous system. Let's remind ourselves of a few key principles. The nervous system is dealing with behaviors, memories, and movement. We said yesterday that the basic functional unit of the nervous system was the neuron, and the neuron had three separate components, or three key components. This was the dendrites, the cell body, and the axon. We said the dendrites were the receptors that received the message. Then from there we said that proteins were synthesized within the cell body as well as neurotransmitters, and the axon was responsible for carrying the action potential. The action potential represented something called electrical excitability, and this is where we're at and where we're picking up. The resting membrane potential is this potential that is due to the differences of ions inside and outside of the cell. So as you can see here, the text says concentration of ions different inside and outside of the cell. And now, what's an ion? An ion is just an element that has a charge. So sodium is Na plus, chloride is Cl minus. So it's the difference in these ions across a cell membrane, which is going to generate a electrical membrane potential. We say here that membrane permeability differs for both sodium and potassium between -50 and 100. The greater permeability for potassium. So inward flow of sodium can't keep up with the outward flow of potassium. This is why we have a sodium-potassium pump. Remove sodium as fast as it leaks in. Here we see a picture that will kind of depict these principles. You'll see the plasma membrane. If you can remember back from Biology 112, you had the hydrophilic heads and the hydrophobic tails in this double phospholipid bilayer. And again, it's the difference in these ions across this gradient, which is establishing this resting membrane potential. You can see here up in the top, you can see that there's an equal number of both positive and negative charges across, this being present extracellular fluid. And here within the cytosol, you'll see also an equal number of positive and negative charges. However, this resting membrane potential is an electrical potential difference across this plasma membrane. It's the differences between here and here. As you can see here, chloride being represented by the negative and sodium being represented by the positive. There's going to be a difference in the amount of sodium that comes across and the potassium that comes across. For every 3 sodium ions that comes in, 2 potassium will leave. Okay, we're going to talk about the principles of the resting membrane potential being set at -70 megavolts. Okay, and then due to an influx of sodium, there is going to be something called depolarization. Depolarization is going to be firing of the action potential. And then eventually we're going to reach something called repolarization, which is going to be primarily due to potassium. So again, these are going to be the key ions that are going to play a major part of this. So here we talk about some other definitions. Graded potential, small deviations from the resting membrane potential of -70. And again, this number -70 is very important. Okay, so far we've talked about the dendrites. The dendrites receive some type of input, and it comes to the cell body. And the cell body is where proteins are synthesized, as well as where the neurotransmitters are synthesized. We have an axon, and the axon generates these action potentials, which are carried down to other dendrites. We said that these action potentials is just due to electrical excitability, right? The electrical excitability is due to the difference in the inflow and outflow of these ions. We said the ions, if you can remember back to Bio 112, ions were just elements on the periodic table that had a plus or minus charge. Okay, so I know this is a lot, but I'm trying to break it down to the simplest way that I can think about this. Okay, so the resting membrane potential is when the cell, right, is most negative. Okay, it's when it's most negative, right? As it becomes more positive due to the influx of these sodium ions, it becomes what we call depolarized, and I'll show you that later on. And then once it reaches back down and becomes more negative again, we call that repolarization. Okay. There's something here called hyperpolarization. Hyper means above. Hyperpolarization means the membrane has become more negative, and depolarization means the membrane has become more positive. The text here says the signals are graded, meaning they vary in amplitude, which is size, depending on the strength of the stimulus and the localization. This is a great picture right here. Very good picture. So as you can see here, the resting membrane potential is set at -70. That means the neuron is not firing. Okay. Whenever a stimulus comes in and raises this resting membrane potential, usually to about -55, we have something called depolarization. This depolarization means that there's opening of the sodium ion channels in which sodium can flow into the cell. Okay. As you can see here, it reaches to about the amplitude of about -60 before repolarization happens, in which the cell is becoming more negative again. So depolarization means it's becoming less negative, which means it's becoming more positive. This means the action potential is fired. And once the action potential has reached, we have something called repolarization, in which it is becoming more negative. Again, here, this is just, if you can remember back to the last lecture, we talked about the different channels that there were. There was the ligand gated, the voltage gated, the mechanically gated. And this is just a picture here showing you, this is the ligand gated. For example, glycine is binding to its receptor. This is allowing the cell to reach its stimulus, right, and depolarization to occur. Okay. This right here is showing you the opening of a mechanically gated channel. Some type of mechanical stimulus, such as applied pressure, has happened, which has opened the channel, which is allowing the inflow and outflow of ions from the extracellular fluid into the cytosol. I like this picture here, just showing you the different stimuluses. For example, in stimulus 1, this has the resting membrane potential is -70. A stimulus has been applied, which has caused depolarization to occur, and then repolarization to occur as well. Depolarization is is firing of the action potential and allowing it to reach a set point, and then eventually it's coming back down. Stimulus 1, this is stimulus 2, this is stimulus 3. But I want you to see this baseline is the resting membrane potential in which nothing is happening. Okay, as you can see, the upslope, this is where depolarization is happening, and then repolarization is coming back down to the resting membrane potential. I like to remember that repolarization is coming back to the resting membrane potential. Okay, they both kind of start with R. That's just a silly way that I like to remember things. Summation is just summation is essentially just the combination of these stimuli kind of amplifying each other. I won't really go into much detail on summation. What is important, and I want you to know, is that this slide right here. So an action potential or an impulse, right? So an action potential, or another word for that is an impulse, is a sequence of depolarization and repolarization. Okay? Depolarization, again, is becoming less negative, meaning it's becoming more positive, and repolarization is becoming more negative, back down to the resting membrane potential. What is causing depolarization and repolarization? It's these voltage-gated sodium and potassium channels. Okay, and I want you to know that action potentials either happen or they don't happen. This is called the all or none principle. Okay, so if a stimulus reaches the threshold, this means that if a stimulus reaches, if it reaches past that -70 to like -55, then the action potential happens. If it doesn't, then it doesn't happen. This is called the all or none principle. This is very important for examination purposes. This is a good picture here showing you that the resting membrane potential is here at -70. This will show you here that we have some type of stimulus that has reached the threshold, which is set at -55, and this is called depolarization. What's happening during depolarization? This is a very good picture and something that I want you guys to know. During depolarization, the sodium channels activation gates are open. Okay, so sodium channels have opened. Okay. We've reached our peak. Now we're back into repolarization. Okay. Now repolarization is due to the potassium channels. Okay. So depolarization is due to the sodium channels which are occurring, and repolarization is due to the potassium channels opening. We talked about um, something called seizures. Seizures is due to abnormal neuronal activation. Okay. So abnormal neuronal activation. Um, Seizures, people have multiple seizures. We call this epilepsy. And um, this is due to spontaneous depolarization, right, of the neurons. So all the sodium channels are opening at one time and causing these neurons to fire. And again, we know that we don't want this to occur all at once, right? This is how seizure activity happens. So just a little real life thing to kind of tie into our lesson. So here, this is showing you the resting membrane potential being present at -70. You have a subthreshold stimulus, and again, we don't have firing of an action potential. Okay, it hasn't reached the threshold. It's all or none principle. And we've reached the resting membrane potential. We don't have anything, and eventually we have firing of the action potential. Right, the threshold has been reached, in which we have depolarization. It's becoming less negative and more positive. Right, and this is called depolarization. Eventually, we're going to have something called repolarization, which is becoming more negative. Right, here's a picture that is very good for you guys to kind of look at it. It'll tie everything in. So during depolarization, the main thing that I want you to take home from this slide is that the voltage-gated sodium channels open and sodium rushes into the cell. That's the important thing. So during depolarization, sodium flows in. During repolarization, the voltage-gated potassium channels open, right? And this will be the last slide that I cover. This is called the refractory period. There's absolute refractory period and relative refractory period. I want you to know that the refractory period is a period in which the neuron cannot generate another action potential. So again, the refractory period is a period of time during which the neuron cannot generate another action potential. The two different types of refractory periods are the absolute, which means there's absolutely no way another action potential can be generated. Okay. The relative refractory period is a period in which there can be another action potential generated if we have a supra threshold stimulus, right? A stimulus above our normal threshold, right? In order to generate an action potential. So this is kind of these are kind of the two refractory periods, right? So we have to ask ourselves, what is a refractory period? Well, this is a period of time in which another action potential should not be generated, right? Or cannot be generated. There's absolute, which means that absolutely there cannot be another action potential generated. And then the relative refractory period is which if we have a stimulus above threshold, then we can generate another action potential. And this is where I'll cover this last slide. Saltatory conduction. So we talked about how the dendrites will receive a message, right? They'll receive some type of stimulus input, and then from there it'll be generated to the cell body and down the axon. We said that down the axon, this is where action potentials are generated. And if you can remember, at the axon, there's something called myelin, right? Myelination. And this is what insulates the axon and allows for conduction to occur at a faster rate. Okay, there's going to be place on the axon called the nodes of Ranvier, right? The nodes of Ranvier. I want you to know that. This word right here, nodes of Ranvier. And this is where there's periods of myelination, right? And this is where conduction is going to occur at a quicker rate. So depolarization only at the nodes of Ranvier where there's a high density of voltage-gated ion channels. So the current carried by ions flow through extracellular fluid from node to node. Okay, I'll show you this. This is the nodes of Ranvier. This is where myelin occurs. This is where myelination is present. And when there is Myelin present, this is where action potential conduction can occur. It's due to the presence of these voltage-gated potassium and sodium channels. Here's a better picture of this. This is the nodes of Ranvier. You can see here where the myelination is occurring, and there's gaps in between here. So this is going to generate a electrical conduction that occurs down this axon here. And this is where we will leave off. So this concludes lecture 2. Again, these are the important principles to take home from this, is knowing that the resting membrane potential is set at negative 70 millivolts. From here, depolarization occurs whenever we reach the threshold for stimulus. Once the stimulus has reached the threshold, the depolarization occurs, sodium ions flow in, and eventually we wind up coming back down to repolarization. Repolarization is whenever we are becoming more negative, and this is whenever the potassium channels are open. And eventually we'll reach the resting membrane potential. We talked about 2 types of refractory periods. We talked about the absolute refractory period and the relative refractory period. An absolute refractory period is a time in which there absolutely cannot be another action potential generated. And we said that during the relative refractory period, there is a period of time in which there's a supra threshold stimulus which can generate another action potential. And finally, last but not least, we talked about the nodes of Ranvier, in which there is myelination that occurs in segments along the axon, and this is where saltatory conduction occurs, which increases the speed of the action potential across the axon. So I know this is a lot of terms, I know these are a lot of definitions, and I know these are different principles than which you are used to. But again, I think just wrestling with the material, going back, reviewing your definitions, and reviewing the picture with all of the slides, this picture right here, I think this would be very beneficial for you guys for understanding what is occurring. Okay. So again, if you have any questions, please feel free to reach out to me. I hope this helps supplement your learning experience. Thank you so much.